The story I'm going to tell you is about a relatively novel connection between skeletal muscle and neurohypophyseal hormones. These neurohypophyseal hormones, as you probably very well recall, are two small peptides, two nonapeptides, very similar nonapeptides, oxytocin and vasopressin. They act on a number of targets, of classical targets in our body through a number of receptors for vasopressin, through a single receptor for oxytocin. Today, we are going to focus mainly on the V1A vasopressin receptor and on the OT receptor for oxytocin. It's important to note, it's important to note that skeletal muscle is not a classical target of either hormone. It's also important to know that in spite of the number of receptors and the presence of specific receptors, however, there are several papers, this is just 3 out of a greater number of papers, showing that there is an important crosstalk among vasopressin and oxytocin at the level of their receptors, meaning that, of course, there are several instances when, for instance, vasopressin activates the oxytocin receptor and causes a specific effect in a given cell type. So we started getting interested in this more or less by chance. We were about 30 years ago differentiation inhibited by TNF. While we were proceeding in our studies, a number of studies came out showing that oxytocin receptors were present in satellite cells. This paper, the first one, refers to human satellite cells, and the OT receptor is expressed, and the consequence of the occupancy of the receptor are again increased differentiation of satellite cells. And these studies were repeated in other species and showing the presence of functional oxytocin receptors in other species. More recently, the oxytocin receptor was reported to be expressed in situ C12 myotubes and to be regulated during differentiation. Myotubes express much more receptor, a much higher receptor level than myoblasts. And above that, estradiol upregulates again the expression of the oxytocin receptor in situ C12 cells, a model of satellite cells. So, of course, so we were facing a number of papers showing that in vitro conditions in culture, we had an effect. I underline the fact that the papers, the data came from different laboratories, not only from ours. But of course, the question was, is it physiologic? So we moved to in vivo studies. This very old paper in 1992 shows that vasopressin, high level of vasopressin measured with different antibodies, 4 or 5 different antibodies, were present in human fetal and neonatal skeletal muscle, then decreased afterwards. We measured the expression of the vasopressin V1A receptor during the development of mouse skeletal muscle, and again we found a high expression at the level at the newborn, and then a decrease after birth. Other papers, more recent papers, showed an important increase in oxytocin expression and the expression of the oxytocin receptor in bovine muscle during development, in particular coincident with active myofiber formation. And a different type of data comes from papers, there are many like this, showing that, for instance, short-term static muscle contraction increases plasma and doubles plasma AVP concentration without concomitant changes in osmolarity, plasma volume, body temperature, all factors that could modify by themselves the vasopressin concentration. You all know very well that skeletal muscle is a dynamic tissue, it's capable of regeneration, and in fact, the mechanical injury and the following regeneration are an important experimental model to study not only development, but also situations occurring in several muscle diseases. And we found that the V1A vasopressin receptor is modulated during muscle regeneration. This suggested us that there could be a role of the signaling elicited by these hormones in muscle regeneration. We also figured that vasopressin has so many physiological targets that it could very likely induce a number of side effects. So we thought we prefer to overexpress the V1A vasopressin receptor in muscle, and in the presence of physiological levels of vasopressin without administering exogenous vasopressin, we verified that V1A overexpression in injured muscle coincided with the earlier restoration of the injured muscle in compared to non-overexpressing muscles. A similar experimental system was that when the injury was followed, muscle injury was followed by administration of not only vasopressin or TNF, and also in this case we verified that vasopressin treatment caused a significant increase in the number of size of regenerating fibers when compared to control anti-NF treated muscles. Also, functional analysis confirmed this type of data, showing that the functional parameters of muscles subjected to damage and then regenerating, the function was deeply impaired by TNF treatment, but almost completely restored by the contemporary treatment with vasopressin. And also, we showed that the overexpression of the V1A receptors attenuates the inflammation and fibrosis induced by TNF, by administration of TNF. A different experimental, different type of problem is that having to do with the changes in muscle homeostasis occurring with age. And this very interesting paper came out in 2015 from the Irina Comboy's group and showed that oxytocin is an age-specific circulating hormone necessary for muscle maintenance and regeneration. In fact, they showed that the levels of OT, the plasma levels of OT decline between young and old mice, that the levels of the OTR receptor show a not so marked, but anyway significant decrease between young and old mice, that the systemic administration of oxytocin to old mice restores the muscle regeneration by improving the function of stem cells obtained from old mice. And they also reported the genetic lack of oxytocin, while not causing a developmental defect in muscle, however, leads to premature sarcopenia. So taken together, all these results tell us that skeletal muscle is, or skeletal myogenic skeletal muscle cells are a target of neurohypophyseal hormones. And in fact, both vasopressin and oxytocin positively regulate muscle cell function or muscle homeostasis in different models, including in vitro differentiation and hypertrophy, in vivo regeneration and aging. Now, in the next slides, I'm going to show you that muscle can be also a source of neurohypophyseal hormones. I'm recalling this paper that I already showed you, showing a very high level of immunoreactive vasopressin in human fetal and neonatal skeletal muscle, but then a number of other papers, such as this that I already mentioned to you before, in 2017, where estradiol upregulates estradiol treatment in C2, C12 cells upregulates not only the oxytocin receptor, but also the expression of oxytocin. And these findings are in line with the number, for instance, of paper that showed along the years evidence of other sites of extrahypophyseal origin of oxytocin. A number of other evidences came from studies performed on farming animals, which are typically exposed to anabolic steroids to increase the meat yield. And in these cases, the muscle expression of oxytocin was strongly stimulated, and this was they report that using a co-expression network strategy, the oxytocin receptor was identified as a major mediator of the muscle response to the anabolic steroids, and OT itself contain an androgen response element, which explains why the chronic administration of anabolic steroids, of course, not only yields increased muscle mass, increased oxytocin serum level, which is probably due to a highly increased muscle expression of oxytocin itself. This is a different paper which shows a very similar thing, and this was done in sheep, where anabolic steroids increase the skeletal muscle expression, and which of course caused an increase in the plasma concentration of oxytocin. Another aspect of where muscle homeostasis is altered, in this case negatively, is cancer cachexia. Cancer cachexia is a very serious muscle wasting condition. It was studied by several people, including Dario Poletti, who introduced this subject in our laboratory, and we mixed the two types of problems, and for instance, very recently, Alexandra Benoni, PhD in our laboratory, set up this kind of in vitro model for cachexia, where the differentiation of L6 cells in culture, of course, is inhibited by the presence of a conditioned medium obtained by the C26 tumor culture. And in this case, the presence of oxytocin reverted the inhibitory action of the conditioned medium. So conditioned medium plus oxytocin was similar to the control, to the oxytocin-treated cells. In vivo, a similar type of experiment in vivo. So here we have a tumor-bearing mice. We have tumor-bearing mice, which show that muscles overexpressing the V1A receptor undergo much less extensive or none at all muscle atrophy, muscle cachexia, which on the contrary is very present, is very clear in non-V1A receptor expressing overexpressing muscle in tumor-bearing animals. Also, a similar type of experiment is again the tumor-bearing mouse, where here the animal is subjected to daily injections of oxytocin, and while in the tumor-bearing animal, the weight of the muscle, in this case is the gastrocnemius, is almost significantly decreased as the cachexia shows up. The injection of oxytocin almost completely prevents the muscle wasting process. This is accompanied by, OT rescues the catabolic genes, which are of course highly stimulated in the presence of the tumor, but not when oxytocin is given to tumor-bearing animals. And I will finish my presentation showing you some very preliminary data obtained in human. First of all, this is in the cancer cachexia context, and it was obtained in collaboration with Marília Silander at the University of São Paulo in Brazil. And these data showed the oxytocin levels in the plasma of oncological cachectic patients are significantly lower than in oncological patients which do not show cachexia. And in another context having to do with sarcopenia, we verified that the expression of both the oxytocin receptor and of oxytocin is much lower in sarcopenic versus the non-sarcopenic muscle biopsies obtained from patients undergoing surgery for a femoral fracture. So the final message of this presentation is that skeletal muscle cells and skeletal muscle are targets of neurohypophyseal hormones, that vasopressin and oxytocin affect the differentiation, the regeneration of different myogenic cell types and of muscle in vivo, that satellite cells and skeletal muscle express oxytocin. The oxytocin expression is modulated by age, by anabolic treatments, by catabolic conditions. And so we can propose OT as a novel myokine. And this leads us to hypothesize that the signaling that goes through the OT receptor may be a reasonable target of treatments aiming to restore ecorrect homeostasis in atrophic conditions such as sarcopenia or cachexia. And this is a partial list of the people who contributed to that part of those results that were obtained in our laboratory. And I thank you very much for your attention.
Sergio, fabulous talk. Any questions? In the brain and having an effect on cognition and also on pain. We have some researchers here at the University of Florida that they're conducting clinical trials. Any indirect effects from that that can relate back, you know, better brain health, less pain, relate back to better muscle function?
Of course, I cannot exclude that. However, of course, the in vitro studies rule out that part of the mechanism that could be very well working in vivo. So it is possible, but not necessarily true. Thank you very much. However, yeah, it's an interesting thing to think about.
Yeah, yeah, very, very nice. I think Ugo's got a question. He's talking. I see his lips moving, but Ugo, you have a question? We have 2 minutes left. We have a question on the chat. Is the expression of oxytocin receptor similar across muscle fiber types?
I'm sorry, I don't see the chat. Can I?
So is the expression of the oxytocin receptor similar across muscle fiber types? Type 1A,
I don't know. I don't know. Okay, the answer is that we don't know.